The overall goal of the following experiment is to validate the use of a luciferase-expressing strain for the non-invasive monitoring of in vivo biofilm formation by Candida albicans cells. This is achieved by implanting catheter fragments colonized with a luciferase-expressing strain of Candida albicans into the back of a mouse. During the second step, the yeast is allowed to grow into a thick layer of cells embedded within an extracellular matrix, generating a mature biofilm on the inside of the catheters. Next, a substrate is added that will be converted by the luciferase enzyme. This reaction produces measurable light. Bioluminescent imaging, or BLI, is then used to record and quantify the formation of the biofilm within the living host.
The main advantage of this technique over other existing techniques, such as the central venous catheter model system, is that we can test up to 6 biofilms inside one animal, thereby greatly reducing the number of animals that we require for statistical analysis.
The implications of this technique extend towards the therapy of Candida albicans biofilm formation, as our method allows the easy testing of existing and novel antifungal compounds.
Also, this method can provide insight into Candida albicans fungal biofilms. It can also be applied to other microbes like bacteria or other fungal species.
The proposed procedures are easy to implement in any laboratory that has access to an animal facility.
"24 hours prior to the surgical procedure, open the package containing